Hello everyone, today I would like to share about fish hematology and blood collection. As introduction, fish hematology, the study of fish blood, is essential for diagnosing disease, understanding overall health, and assessing environmental impact on fish. Blood tests help us identify infection, nutritional deficiencies, and other health issues that might affect fish population. Collecting blood from fish requires specialized technique to minimize stress and ensure accurate results. This involves using specific tools like syringes and anticoagulants. Proper handling and sampling methods are crucial to obtain reliable data. By examining blood parameters, we can gain valuable insight into the health of fish. This knowledge is vital for maintaining the health of fish in both aquaculture and natural habitat. It helps us promote sustainable practice and conservation effort, ensuring the well-being of fish population. The material used in fish hematology and blood collection and their function. First is live fish. They are the subject from which blood sample will be collected. Second is anesthetic solution. This is used to anesthetize the fish to minimizing stress and movement during blood collection. Third is sterile PBS or phosphate buffered saline. This solution is used to rinse and maintain the sterility of instrument and sometimes to dilute blood sample. And fourth is sterile syringe or 1 ml or appropriate size. Syringes are used to draw blood from the fish. Next, we use sterile needles in 23 until 25 gauge or appropriate size. Needles are attached to syringe for blood collection. The size of the needle ensure minimal harm to the fish. 6 is anticoagulant. For example, we use heparin sodium citrate, potassium ethylene acetic acid or EDTA. These substances are added to blood sample to prevent clotting, ensuring accurate hematological analysis. The next material we use is microscope slide and cover slips. They are used to prepare and examine blood smears under a microscope. And next is tubes used to collect and store blood sample. 9 is hemocytometer. This instrument is used to count blood cells, providing critical data on the fish health. Other than that, we use microscope, essential for examining blood smear and performing detailed hematological analysis. 11 is Reich stent or Giemsa stent. These stents are used to highlight different components of the blood on slides, making it easier to identify and count cells under a microscope. The next material we use is disposable glove and lab coats. These are essential for maintaining sterility and protecting the user during blood collection and analysis. Next is a centrifuge. A centrifuge is used to spin micro-centrifuge tubes at high speeds. This process separates the different components of the blood, such as plasma and cells, allowing for hematocrit determination and other analysis. It is crucial for measuring the proportion of red blood cells in the blood. The last one is hematocrit reader. A hematocrit reader or a ruler with calibrate marking is used to measure the packet red blood cell column length accurately after centrifugation. This measurement help determine the hematocrit value, which is the percentage of red blood cell in the blood, providing essential information about the fish health and oxygen carrying capacity. These tools are vital for precise and accurate hematological analysis, contributing to our understanding of fish health and aiding in the diagnosis of various conditions.
Step 1 is fill the container with water from the fish tank and add the appropriate amount of anesthetic solution, which is insert 2 drops trans more. Then gently transfer a fish from the tank to the anesthetic solution and allow it to anesthetize and ensure the fish fully sedated before proceeding. After that, we using a sterile string and needle and carefully punch the caudal vein and gently draw the blood into the string until 50 to 100 ml. After that, we put a small drop of the collected blood near one end of the slide and using a second microscope slide held and spread the drop of blood along the length of the first slide to create a thin smear. Allow the smear to air dry completely. And this is look after air dry. Then put the blood in the tube. Other than that, the heparin microhematocrit is inserted into the blood sample until it fills approximately 3/4 of the capillary tube, and one end of the capillary tube is blocked by sticking it with the wax plug. Then we centrifuge the capillary tube for 5 minutes using microhematocrit centrifuge at the speed of 1500 rpm. This is for isolate the blood cell and plasma.
And as for the blood collection result, we managed to ensure that the volume collected is sufficient by using the 1 ml syringe full. And we also calculated the amount of both red blood cells and plasma cells by using the hematocrit reader. The hematocrit value was examined using the microhematocrit method, and the result is as shown in the table. We have two samples of the capillary tube, and as for the result, for both sample 1 and 2, the total of hematocrit is 7.5 mm, and for the red blood cells for the sample 1 is 0.3 mm and 0.4 mm for the sample 2. The amount of plasma cells in the sample 1 is 4.9 mm and 4.5 mm for the sample 2. And for the percentage of the hematocrit for sample 1 is 0.04%, and for the sample 2 is 0.053%. For the staining result, we examine the stain smear under a microscope at various magnifications to observe and identify different blood cells types. For example, red blood cells and white blood cells. So based on the observations, we managed to identify the plasma white blood cells and the general characteristic of it. White blood cells are typically stained using the Reutz or Giemsa stain for microscopic examination. This staining highlights the nucleus and cytoplasmic granules, aiding in the differentiations of the white blood cells types. Most white blood cells are roughly spherical when suspended in blood, but may change the shape when they move out of blood vessels and into tissues. So for the plasma characteristic that we managed to observe through the microscope is the plasma cells are large lymphocytes with abundant cytoplasm and a characteristic appearance on light microscopy. They are round to void cells containing abundant deep blue cytoplasm with a pale perinuclear area corresponding to the Golgi apparatus, and they have a round, eccentrically placed nucleus with coarse chromatin arranged in a clock-face pattern. So in conclusion, in this blood collecting and smear laboratory practical, we successfully demonstrated the essential techniques for obtaining and preparing blood samples for microscopic examination. By carefully collecting blood and creating a smear, we were able to observe and identify a white blood cells and with its distinct characteristic. This practical exercise reinforced our understanding of the morphology of the white blood cells and the importance of proper sample preparations in diagnosing and studying various hematological conditions.